Welcome. Today we're moving beyond check-the-box compliance. We are looking at process validation as a scientific life cycle, a continuous journey that bridges the gap between pharmaceutical development and patient safety. This is based on the 2011 FDA guidance, which shifted the industry from a static event to a dynamic system of proof. Forget the rule of three. The old mindset was 3 batches and you're done. Today, the FDA defines validation as the collection and evaluation of data from the design stage through commercial production. It's an infinite loop. Every batch is an opportunity to gain continuous knowledge and scientific evidence of consistent quality. This isn't just a best practice, it's the law. Under 21 CFR 211, quality, safety, and efficacy must be designed and built into the product. You cannot simply test quality into a drug by inspecting the finished box. If you haven't validated the performance of processes responsible for variability, you aren't just out of sync, you're out of compliance. We break this life cycle into 3 distinct stages. Stage 1, process design, where we define the how based on development knowledge. Stage 2, process qualification, where we prove the process is capable of reproducible manufacturing. And Stage 3, continued process verification, where we ensure the process stays in a state of control during routine production. These stages are integrated. Data from one fuels the success of the next. Stage 1 is about building process understanding. We use design of experiment, DOE, to reveal multivariate interactions between variable inputs and outputs. While these early experiments don't strictly require cGMP, they must follow sound scientific methods. The goal? A master record built on risk analysis, not guesswork. How do we handle variation? We either reduce input variation or adjust for it during manufacturing to assure consistent output. This is where process analytical technology, PAT, shines, using real-time analysis and control loops to adjust conditions dynamically. If your product attributes aren't easily measurable, like viral clearance or microbial contamination, this controlled strategy becomes your primary line of defense. Now we scale up. Stage 2 has 2 parts. First, we qualify the facility, utilities, and equipment, including installation qualification, IQ, and operational qualification, OQ. Then comes the PPQ, process performance qualification. This combines the qualified facility with trained personnel using the actual commercial process. Before you run a single PPQ batch, you need a written protocol specifying manufacturing conditions, controls, and expected outcomes. It must include a sampling plan that provides statistical confidence of quality within and between batches, usually much more extensive than routine testing. You need clear, science-based acceptance criteria and a rock-solid plan for how to handle any data that doesn't fit the mold. Execution is critical. Follow CGMP-compliant procedures and use personnel routinely expected to perform the operations. The final report must be transparent. It summarizes every non-conformance or deviation and must state a clear conclusion. Is the process in a state of control? Without a yes, you do not commence commercial distribution. Validation doesn't end at launch. Stage 3, or CPV, is about ongoing assurance. We use 21 CFR 211.180E to collect and analyze product and process data from routine manufacture. The goal is to detect unplanned departures from the design and identify problems before they become product failures. This is where the statisticians take the lead. We use statistical process control, SPC, to monitor intra-batch and inter-batch variation. We aren't just looking for passes or fails. We are guarding against process drift and unintended variability. If you don't understand your noise, you'll never see the signal. The life cycle is a feedback loop. Complaints, out-of-specification (OOS) findings, and operator feedback all flow back into the system. If the process drifts, we investigate and adjust. Remember, significant changes require a justified rationale and may mean going back to stage 1 to redesign or stage 2 to requalify. Your process knowledge is only as good as your yardstick. While development methods just need to be scientifically sound, any method used for commercial batch release must be fully validated under CGMP. Better technology means better understanding, and the FDA encourages that evolution. Transparency is the heart of the scientific method. Stage 1 captures the why behind design decisions. Stage 2 involves heavy CGMP documentation like protocols and reports. And Stage 3 maintains trending reports. Process flow diagrams are your key tool here, mapping unit operations and control points across the entire life cycle. To wrap up, process validation is the bridge between pharmaceutical development and patient safety. We strive for homogeneity within batches and consistency between them through scientific evidence of control. Why? Because at the end of the day, quality is designed in, not tested in. That is the science of compliance.